Okay, well, here's Bradley.
Hello.
Ted was just explaining to me that he just joined DevoWorm.
Ah, hello.
So,
hello, Bradley.
Hello. How are you?
I'm fine.
Yeah.
Actually, I'm pretty fresh here, so I just joined DevoWorm yesterday, so I'm trying to follow up our works to see what can I do here. So I want to attend the weekly meeting to listen to get some information.
Yeah, that's good. Well, we have an archive of meetings, so and we're right in the middle of our Google Summer of Code. So we're reviewing projects on a weekly basis and going through some of the things that people are doing for their Summer of Code activities. Who's clicking? What's the clicking coming from?
Okay.
Okay, thanks. So the Google Summer of Code activities, and here's one of the people from Google Summer of Code right now, Harikrishna. So, but we have an archive of meetings, and in some of those archives, I'll respond to you in Slack. We'll have, we have a YouTube channel, we have some data sets, and I have an onboarding guide I can send you, and that should start you off on some things. Just take a look around and see what interests you, and if something during the meeting interests you, don't hesitate to, raise your hand, and you can use the chat, or you can unmute yourself and ask about something. Yeah.
Okay, thank you very much.
Yeah, no problem. So glad you could make it. Harikrishna, how are you?
Yeah, hi.
Hi.
Yeah, I'm fine now. I just came back from college 1 hour before.
Oh, good. Okay. Do you have an update for this week?
Yes. So, in the last meeting, I mentioned about the algorithm which I tried. So, I wanted to show the result from which I got using that.
Okay.
Actually, I tried to use the Embryo dataset, but during the algorithm showed the error saying that the images are not much continuous. So for the sake of getting a result, I tested out a different 360 degree view data from the internet, and it gave an output. So I'm now trying to make it work for this data set. So I'll show you the result, what I got from those data sets.
Okay, looks good.
Just a minute, I'll just open.
Are you going to share your screen, or?
Yeah, I'll share it and show it now.
All right. Okay. You're muted, Harikrishna. Yeah. Yeah, okay.
Yeah, it's visible, right?
Yeah, I can see it.
So I'll show you the data set first.
Okay.
So I tried using this images to generate the 3D model.
Oh, wow.
It covers all the area 360. So the result it gave is, I think it's very nice. Like, if you see.
Yeah.
Yeah.
So this
is a complete 3D model.
Is this like the model and then you're putting it in the background, or what is this? Are you taking the vertices out, or?
It's the 3D model, the object file of those, which is created from the algorithm, the output file.
Okay.
Yeah. So
Good.
what I'm trying to do now is to create a similar model from the embryo images.
Okay. Yeah. Yeah, that looks good.
I don't know will it work or not.
Okay. You said that you had a problem
I
with
even asked
discontinuous. Oh. You said you had a problem with the discontinuous files or something? What does that mean?
Yeah, when I tried using the embryo dataset, it showed that the images are not very continuous. I guess it needs more continuous images. I guess. I'm not sure.
Yeah.
Try some image processing on the images to make
Yeah.
them more, do they stand out more? More
Yeah,
contrast?
that's what I'm planning to do.
Yeah. And are you using some from the 360 images that I sent to Sync?
Yeah. I used from
Yeah.
that.
Because there's some there that are more complete, like they have more sides showing. So,
Okay.
yeah.
I'll test out everything.
Okay, I don't think I have any that show all of the sides properly because a lot of the bottom images got blurred. But anyways, I can look some more on that, my other computer for the images where they're stored. See if I can find some more.
Okay.
Okay. That looks good, the pinecone example. So you're going to try to build an embryo model of this
Yeah.
next. Okay. Well, it's good. Good, thank you. Karan, how are you?
Hi, Bradley. Hi, Susan.
Hi.
Yeah, things are going great. Like the weekly schedule is holding up. Nothing too ordinary out of that. Otherwise, Harikrishna, I think he mentioned he's using ColdMap, so that was pretty interesting as well. This week's task was trying to finalize how many embryo samples I'm going to be using. So far, I think from stages 3 to 5 are kind of okay. So I'm still thinking of ways to incorporate more stages of axolotl, because like I'll share the drive link first.
All right.
I'll share it in the meeting link. So the difference between those images, I'll either have to incorporate another step, to unblur those images, especially when you get to stage 6, stage 7, and stage 8 axolotl embryos, right? The number of cells and it gets very hard for the algorithm to decide, to find corresponding points between two image sets. Like if we have it for, wait, let me just share the link first. I'll share the Google link drive. I'll just try to find it.
Okay.
Have you seen the 360 images that I've got on Sync, Karan?
I think I have more than those. You'd sent me, Susan, do you remember you had sent me, you'd uploaded some images on the Google Drive link that I had sent
Yes.
a while back, yeah. So I've mostly been using those only. I think I might not have are they more recent than that, or?
Yes, I uploaded all of the 360 I'd found that were good onto Sync.
Okay. I think Harikrishna may have sent it to me. I'll just check.
Richard Gordon Sync.
Okay.
Under my name. There.
Okay. I'll just check.
Okay.
Yeah.
All right. Oh, so was that your entire update, or?
Yeah. I was actually trying to find the Google Drive link, to
Okay.
describe the difference in that. But yeah, this is it. So I'll probably try to find just to make those images more usable. I'll try to find ways to do that.
Okay.
There are a lot of computer vision algorithms that try to unblur images without losing a lot of detail. So, or adding in too many additional things. So that is one of the things. So something along those lines maybe. But otherwise, for stages 3 to 6, there's at least it won't give too many branching options as to way to go about when we have to finally get the final model. Yeah. So yeah, that is there. Otherwise, I think this was the main thing. Otherwise, I keep on checking alternatives for 2-3 of the things like I had mentioned. Especially the second part where we have to generate the 3D model from the 3D point cloud. So for that, I've not found any better alternatives so far. So I'll be sticking with my own method. That's there. And then also about the Mars shot thing, I'm still searching for a neural net that can create a 3D probabilistic point cloud for doing some of these things. So I think instead of searching for things of this sort, I think I'll have to take a very basic boilerplate code for doing something like this, and then modify it specifically for the axolotl model. So that is also something that I was looking into. Otherwise, for the weekly goals, these were the things that I had. The main part of it was just finalizing which embryo stages I had to choose, and that I'll be going forward with properly. So that range so far is 3 to 6. I'll try to increase more, if I can find a better way to use more images for that.
Yeah, I would work on getting it kind of debugged first, and then the secondary images would come later. But you'd know at least the basic steps of how to do it, and then you could work out some of the image processing problems.
Yeah. I'll look into it more.
Yeah. Well, that sounds like we're coming along good, and both Harikrishna and Karan are working on this. And they have their own. Looks like they're going in complementary but different directions, so that's good. And then from Jiahong and Wataru, I've been talking with them a lot on Slack, and they're working on. They're making some progress on their work. We had some discussions about, I think Jiahong is interested in topological data analysis, something that we've talked about in the meetings, but not really kind of dug into very much. So he might be going in that direction. But yeah, they're working on their projects as well. And I don't, it's a little late for them when they do make the meetings. I think it's like 11 at night for them. So, but I'm gonna share my screen. I wanted to go over our GitHub repository. So this is the GitHub repository that we have for GSoC, and it looks like Karan has made his first contribution to this. So he's put in a README and initial draft Python file here, and then some write-ups. So this is kind of what I was thinking about for submitting things to the GitHub repo. So if you're making I'd like to see maybe at least regular updates, if not every week. And you don't have to make a pull request, but you can, because I think all of the students have write privileges to this repo, so you can just put things in here if you want. And then I know that, so this is Karan's update here. He's putting in details. He's got a Python file, so this has some code in it. And then he has this README, which, you can put in. We can do this later, actually, if we want to build a README, because when we have the finished product, maybe a README would be good to have. Well, we have to have a README for the submission to Google, but, that's part of it.
Yeah.
Or are you going to say something?
I'll update it. Yeah.
Oh, okay, yeah. And then the write-ups, which, if you want to create write-ups, I had asked people, I think, if there was a requirement this year for doing like a weekly blog post update, and I don't think there is this year. But in past years, they've done like weekly updates on people's progress. Now you don't have to do that, but it's nice to keep a little diary of what you've done. So in Karan's case, he has this write-up for community bonding period, introduction, and then kind of describing what has been done. And, this is a nice, again, you can go back to this at the end of the project and see what your progress was. So this is, writing this up in the moment and then looking back. And so, yeah, week 1 here, he has some points, some bullet points that you can follow up on. And so and then I did talk to Jiahong and Wataru about working on theirs. And they're going to they're going to work largely from their own repository. So I'm not really sure how to get to that repository. It's actually in Jiahong's repo. But I don't know. Yeah, I don't know where they are on that. But I actually looked at it, and they do have some code up there. So in lieu of me going through that code and not understanding what they're doing, I'll let them do it let them update us at another time. But I just wanted to go through that GSoC portion so that people were familiar with what how that would work and how we're going to go forward on that. And you can work from your own repo. It's fine. I just wanted to have people give an update maybe once a week or once every 2 weeks, so that we can keep things, we all have kind of a common place where they're going to be, where at least, you don't have to put every file you have on your own repo up there, but just to have like a record of it. Okay, do we have any questions at this point, or?
Yeah, I'll be updating the README. I think with portions of how to go about installation maybe when we have the final package ready, or a useful helpful link as to how you can interact with the model and all. When we get documentation for projects and all, something along those lines.
Yeah.
Yeah. So I think I'll at least start with a rough draft, so that even people who are being redirected to the project right now at least have something to get a basic gist out of it, rather than straight away going with the documentation after finishing the project.
Yeah. Yeah, a lot of times though, it does help to keep like the documentation in parallel with the project, because you can't always remember what you did sometimes.
Okay. Yeah.
But yeah, it's good. All right. So that's good. Did anyone want to present anything, or do we have any other things we would like to share before we move on?
I have an interesting paper that I'll send you, but maybe that's for next week. It's how microtubules produce electric charge.
Oh, wow. Yeah, that would be good if you could send that along.
Yeah, I'll send it for later. Bundles of brain microtubules generate electric oscillations. That's the title. So I'll get it to you for later.
Yeah. That sounds great. I'm just making a note here. All right. So I'm going to move on next to some things that I have to share here on, I have a couple things. So the first thing is that we have our submissions document for 2022. This is our list of things that we have outstanding that we're submitting to different places, and perhaps things that we want to put on the list to target different venues or things that are sort of fallen by the wayside and we can pick them up again. So to go over what we have here, we have so in green, these are things that have already been presented or accepted. So I have I'm going to be actually at NetSci 2022, which is the network science conference. And, I'm going to be participating in both the main conference and the satellite sessions. Main conference, I'm going to give a talk on divergent integration of embryo networks. So this is a thing that we've talked about in the meeting with respect to embryo networks. The divergent integration aspect of it is just where you have the formation of different networks based on different parts of the embryo differentiating. So when the connectome differentiates, for example, this forms a subnetwork with respect to the rest of the embryo network. And you start to get this divergence of subnetworks over development. But they're also integrated because they're part of the same organism. And so this kind of described the network theory terms what that process looks like. So this has actually been, this is the accepted one here. I have this, I guess, twice. I don't know why, but in any case, we have, I'm going to present that next month. It's coming up. So that's actually green. I should just put green on that. I'll fix this later, but I just wanted to go over some of the things. The next one is this test of Williamson symbiosis, and this is something that Dick Gordon has been championing, and this is a paper on looking at different genomes. It's a bioinformatics heavy thing that if you were to do this, looking at different genomes and looking for evidence of overlap in developmental programs, and we'd have to go through what that is a bit more, how that process would work. But it's basically looking for the genetic precursors of symbioses and what they call compound development, which is where an organism goes through different stages of development. Sometimes they change their phenotype quite drastically in development. And like caterpillars, they start out as they're crawling along on tree branches. They go into this cocoon, and then they emerge as a butterfly. And that process involves a lot of de-differentiation of cells and redifferentiation of cells. And so there's thinking that there might be some precursors in the developmental program that allow for this. And the test would be to look at genomes and compare their structure and their content. So that's something that is a longer term thing. We've never found anyone who wants to do that and pick it up and move that forward. But that's something that we have outstanding. We also have this Diatom movement project, and this is looking at these. We do work with diatoms, so we do work with these. They're single cell organisms. Sometimes they exist in single cell form. Sometimes they exist in colonies, and we're interested in their movement. So we've done a lot of image processing on images of diatoms. We have access to libraries of diatom images, and we want to look at the movement and track the cells and see if their movement is smooth, which means that it proceeds in a straight line, and that when it curves, like in colonies, sometimes they oscillate. So if the oscillators are smooth with respect to their endpoints and across their range of motion, or if they're jerky, meaning that they exhibit like very high frequency oscillations, sub-oscillations. So in movement, they call that jerkiness because there are a lot of higher order derivatives that you can, if you apply higher order derivatives to the data, you can gain a lot of information about some of their movement behaviors. So sometimes if you have like a stochastic motion where it's just generated by a random process, that jerky movement, you can pick up jerks in the movement trajectory, meaning that it's not smooth, it kind of oscillates along a line. And so this is the kind of thing we're interested in. And you can derive this from image data. You can derive this from microscopy movies that people have made. And, some of the movies available are at a low sampling rate, but we have movies that are of a higher sampling rate that we can use for this. And we can compare those movies, and you can do a lot of mathematical transformations of the data. And so this involves a number of things. This involves image processing. This involves mathematical modeling, and it involves comparative work, because you might want to look between different types of diatom to get a sense of what we're dealing with in terms of the movement, to interpret the movement. So there's a lot of work there. We've done work on diatoms previously. So if you go to our GitHub repository for DevoWorm, we have a project called Digital Basilaria. And this project started a couple years ago. We've had a number of contributors to this, and the idea here is that we're trying to model these. So these are diatoms. They're marine microorganisms or algae, and they exist in these colonies, or they can exist as freeform single cells. Sometimes they have, they're shaped in different ways, so they have sort of rounded phenotypes. These are elongated phenotypes. So these are different, but they're arrayed. In this case, they're arrayed in a colony, and they move against one another. And that's sort of why this organism is interesting, because they have this unique mode of movement. It's like an accordion. And so this is something we want to interpret. These are the species. So they exist in these accordion-like colonies. They exist in these chains. They exist in different forms. And so it's not very well known. The diversity here is not very well known. There's some diversity that's well known in terms of like people studying certain species, but the movement is definitely not really well understood. There are a lot of competing theories as to why they exist, why they move the way they do. So this is part of this project. So we're not just doing nematodes, we're doing other organisms. There's also some work that's being done on archaeobacteria and looking at how their shape can be compared to the shape of oil droplets. So oil droplets, of course, are inorganic, and archaeobacteria are bacterial, so they're living cells. So what are the differences between those two? And that, again, is something that involves image processing. We're also interested in doing work with cellular automata and what they call Wolfram Patterns. So this is I don't know if people are familiar with the book A New Kind of Science, but this is a book that came out about 20 years ago by physicist Stephen Wolfram. And he argued that you can take these cellular automata, these grids of cells that change their state as they interact, and you can classify the patterns that emerge from these. So if you run a cellular automata for a long enough time, they generate different patterns. If you apply different rules to the interactions of different cells, you get different patterns, and you can describe those patterns. And what we're kind of interested in is, are these rule sets closed or are they open? In other words, can you produce things that look like biological patterns through a closed set of rules, meaning that there's like just a finite set of rules that always apply and always produce that same pattern? Or do you need new rules occasionally to maintain those patterns? And so, there are a lot of, we would need to talk about this if someone wanted to do this. This involves, I think, working with a simulation package, working with like cellular automata, but also maybe some image processing of patterns. If we have images of patterns that we can decompose and find some of maybe the rules of some of these patterns as they form or as they exist. There are a lot of creative solutions to this, and I don't really know what they are. We talked about doing like a 1D Ising model. So there's, that's something that we haven't really moved very far on. So if people are interested, this is something that we have. This is the DGNNs. This is the developmental graph neural networks. This is the project that Jiahong and Wataru are working on. And we want to create graph neural networks of developmental processes. So we have these embryo networks, which are cells in an embryo that form networks. They could be proximity networks, they could be signaling networks. And then taking those networks and finding embeddings, taking the data from actual embryos and finding graph embeddings that describe those embryos. So the positions of cells, the positions of different things, structural things in the embryo, and then building graph embeddings to describe that. And then our target here not only is the end of GSoC and successful end of GSoC, but there's Learning on Graphs conference. And so this is September 9th and 16th. So this deadline, I think this is September 9th is for the abstract, 16th is for the paper, I believe. We have to write a paper up, yeah. And then we have, so this is a nice end target for GSoC. But if other people want to be involved, we can talk about it. Jiahong regularly talks about this project in the group. So if you want to go back into our archives of meetings and find the talks that Jiahong, or find the things that Jiahong has talked about on this, or, stick with us through the summer, we'll be talking more about this. Then, NetSci has a number of satellites. So I'm going to be presenting at NetBiomed 22, which is a satellite of NetSci. And this is on a different topic than the divergent integration. This is on hard to define events in biological networks. And so what are hard to define events? I did a workshop about 10 years ago on something called hard to define events. And these are things that are hard to define. They might be like rare events that almost never happen. They might be, hard to describe measurements. So, you might be dealing with something that you can't really measure directly, and it's very hard to get a grasp on what the measurement looks like. Other types of things like non-normal distributions. So things with a very long tail. And so these are all these hard to define events. And what I'm going to do in this talk is I'm going to review what we did at that workshop. And I also did some follow-up. I used to run a sort of an annotated list online for about a year after the workshop. And I would add in things that looked interesting. So there are all sorts of mathematical approaches to these kind of problems, all sorts of different their approaches using network theory, but no one's ever really integrated them into under the same topic. And so that's what we were doing back then. Now I want to review those materials and bring them forward into what's going on in network science today. So I'm going to present on this when I'm done with the talk, but it's this is something that I'd like to resurrect, this idea of hard to define events, but I'm not really sure how. But that's okay. I think it's really relevant to what we talk about in this group. So more on that later. Finally, we have this differentiation tree of the brain, which is where Dick Gordon has derived a lot of his work on differentiation trees and differentiation waves. And it involves looking at our lineage trees, or looking at like the line of descent in an embryo in a little bit different way than we're used to. So we're used to what we call lineage trees, which are trees that branch, cells divide and you get cell lineages. So one cell has, one mother cell has two daughter cells, or maybe a collection of daughter cells, depending on the type of embryo. And then you can trace out, different identities for these cells. With a differentiation tree, it's a little bit different, because you're talking about, the differentiation of tissues and the tissue precursors. So for example, if you have a population of precursor cells, those precursor cells will give rise to maybe muscle cells, and brain cells. And then those brain cells and muscle cells will, branch off into not really sublineages, but subgroups. And so you end up with this tree of differentiated tissues. And so this is something that we were talking about doing. This may need to be refined, but I want to bring back, we've done some papers on this work in the past. I want to turn our attention back towards this a little bit more. And so I want to go over maybe some examples of this. So this is one example of a differentiation map here. This is where we have, this is from one of our papers, actually, a figure. And I was asked, Jia Hung asked me about this, so I want to bring it up. So this is sort of in C. elegans, this is how we envision a different, I call a differentiation map. And it's a differentiation tree in C. elegans, but it's embedded in the embryo, and it's at least two-dimensional topology on the surface of the embryo. So this is the anterior to posterior axis, or this is the anterior to posterior axis. This is the left to right axis, I believe, or maybe I have that backwards. But anyways, this shows like the positions of the cells, and then the direction of differentiation. So if we want to go down the AB lineage, so in C. elegans, the differentiation tree and the lineage tree are pretty much indistinguishable, for reasons that because of C. elegans having a deterministic mode of development, where every cell, what its fate is going to be. So, but there are differences. So this shows like this differentiation map where you have going from P0, which is the single cell state, to AB and PL, or AB and P1. So these are this is a two cell state. And then you go down these different lineages. So you have AB, ABA, ABAL, and then down to ABAR. Now, what are these green and red arrows? The green and red arrows represent different asymmetries in the cell size. So when you have a division, generally, you might assume that it's a symmetric division where it's roughly about the same size, 50-50. So you take your mother cell, it has a certain volume, and the daughter cells, if they're symmetrical, will be roughly half of that. So it's like half of that size. And so then, this continues down the way. But in a lot of cases, you have asymmetric divisions, which are where the size is different. So it could be that your daughter's, one is 60% of the original mass of the mother, one is 40% of the mass of the mother. That's considered an asymmetric division. So this differentiation map actually shows that they're different, they're different paths for these different types of cells, larger cells and smaller cells. And so that's what these green and red arrows are. And they go in different directions. And you can see that you not only can get that information out of, and this is from cell tracking data where you were able to calculate the volume of the cell. And it wasn't a perfect measurement of volume, but if you're using good markers for the membrane around the edge of the cell, you can get a pretty accurate measurement of size. And so these size differences are important. So that's why it's important to have not only sort of a centroid position when you're doing cell tracking, but sort of a volumetric measurement as well. And there are different ways you can approach volumetric measurements. So the difference between lineage trees and differentiation trees are as follows. So for lineage trees, we have a mother cell and we have two daughter cells. Sometimes we have multiple daughter cells if you have cell proliferation, usually not in C. elegans, but in other species. Then one of those daughter cells in turn becomes the mother cell and gives birth to new daughter cells. And again, this can be a proliferative process where you have more than two daughter cells. But for the case here, we're just kind of interested in the C. elegans lineage tree, where we have two daughter cells per mother cell. But as you can see, this creates a tree-like structure that has both sort of heredity and it has this sort of sublineage definition, so that this is a sublineage of this tree, this can become a sublineage of the tree and so forth. With the differentiation tree, it's a little bit different. So in the C. elegans case, we might be interested in a differentiation tree, and in that case, it's going to look very much like the lineage tree. It's going to have the same tree structure, and each of these nodes is a cell. But we're interested instead of in the heredity, in other words, the mother cell giving rise to two daughter cells, and then this axis being the anterior-posterior axis. And in different species, it's defined in different ways. Sometimes there's radial symmetry, in which case these endpoints of the tree correspond to different parts of the symmetry. So in C-squarts, there's a fourfold symmetry. So some of these subtrees define like one part of that symmetry. And so you have four subtrees that are basically very similar. They represent each of those four folds of symmetry. And the differentiation, so this is the lineage tree. This is the differentiation tree. And so here you have a differentiation tree with cells that divide, and they have mothers and daughters just like this. But we're interested in the difference between these two cells. We're interested in the difference in their volume. And so now, if we want to have the smaller cells to the left, the larger cells to the right. So we order these by the volume of the daughter cells. So these the tree is organized a little bit differently. These nodes get flipped around, and so this order is no longer AP, anterior, posterior, it's volume. And so this reorders our tree, and it tells us something about differentiation throughout the tree. Now in a regulative embryo, which is not C. elegans, so C. elegans is actually a mosaic embryo, and you can use this method for most mosaic embryos. For a differentiation tree in a regulative embryo, you still have this tree-like structure, except instead of cells, now you have tissues. And so now you're differentiating different tissues. The reason why the lineage and differentiation trees in C. elegans is basically the same is because we're really, in a C. elegans tree, all the cells are deterministic with respect to their fate. So, every one of these sublineages has a certain set of cell types that will emerge from it. Every cell has sort of an identity, so if you go through this lineage, you'll end up at the end of development with cells that have specific functions. We have classified all of these, they have a nomenclature for this, and they're precursors to defined cell types. And it's very hard to change those with cell signaling or with environment. They're basically there. In a regulative embryo, cells will branch like this, and their fate will be dependent on where they are in the embryo and the signaling mechanisms that they get exposed to. So in C. elegans, in this mosaic form of development, it's basically the same as the lineage tree, except it's reordered in a regulative sense, a regulative embryo. These nodes are now groups of cells that function as independent tissues. And so here you have precursor cells. Here you might have like a muscle fiber or a set of muscle cells that might start to differentiate. And then here you might have different types of muscle, different specific muscles in the emerging embryo. So you can see that there's this differentiation tree, that things are differentiating. These might be neurons. This might be like the neurons in the head, the neurons in the tail. Okay. We don't necessarily do this in C. elegans because we don't have to. We know what the fates are going to be, so we can actually, and there are other methods that we can use for this. We're working on a categorical theory methodology for this, a sort of a hybrid hypergraph and a category theory method for this. But it's different than this sort of differentiation tree where when cells differentiate into a new tissue, they get a branch. So you can follow back these signals from the very specific types of cell tissues that get generated to a very general set of cell types, or a very general population of cells that emerge. And this is important when you have different regions of the embryo forming tissues and maybe cells changing their fate as they're exposed to different signals. And then there are things called differentiation waves, which are waves of differentiation that occur in space. And so that's something we can talk about at another time. But this is just a little sketch of the difference between lineage trees, differentiation trees, in two modes of development. Dick's here. Okay, hi.
Hello. Running way behind today.
Oh, it's okay. So not only do you have these type of divisions like visualized in space, and you can see that this is the lineage tree. So the lineage tree just tells you a line of descent. This tells you a lot more information. The other piece of information you can get out of this is an angle of differentiation. So if you have like cell tracking data and you have like the mother cell's position and you have the daughter cell positions, you can interpolate, and you can have this in 3 dimensions. So you can interpolate an angle at which differentiation occurs, and so or division occurs. So you have the AB cell, it divides into ABA and ABP, ABA going towards the anterior end or the head, ABP going towards the posterior end and towards the tail. And then there's this angle of division that is maybe important. We don't really know. I don't think anyone's written about this as a paper, but this is something you observe in these maps that show great variation in the angle. So this has a pretty wide angle here between ABAL and ABAR. ABA and ABP have a little bit smaller angle, and then AB to ABA, ABP, that has a different angle. And so you can like calculate these angles out. Sometimes they're almost 180 degrees. Sometimes they're a lot narrower.
Bradley?
Yeah.
Has anyone checked as to whether or not, think about being in a room surrounded by other cells.
Yeah.
And you're trying to double yourself and get out of that space. Okay?
Yeah.
There might be a preferred mechanical direction for squeezing out of the space. Has anyone looked at it that way?
I don't think so, but that sounds interesting.
Yeah, it's sort of think like a cell.
Yeah.
Okay. What's the easiest way out? Does that determine the angle?
Yeah, right.
Okay, now that might be, you'd have to sort of do local strains of markers, of some kind of markers to see if you could figure out what the direction of minimal strain was, or easiest way to double.
Yeah.
Because after all, when a cell doubles, it's also going, each daughter cell may or may not go back to the original volume.
Right.
Especially if it does increase in volume, then it's got to squeeze somewhere.
Yeah.
So can you, how difficult is it to get the volume tree? In other words, the volume of each cell versus time along its tree, along its edge on the tree.
Right.
Is it possible to determine those volumes?
Well, like I just said before you joined us, we can estimate it from from imaging. We can use image processing to, if you have really good markers for the.
Okay. Because in mammalian embryos, the size starts out very large but keeps decreasing, and I'm not sure if anyone's tracked how far down it goes. Or when it stops decreasing.
Yeah.
I don't know anything about nematode cell size.
Yeah. Well, they do. Yeah, they do. Like when they divide, upon division they have half of the original cell size, or maybe a little bit of difference there. But then I don't know how much they increase in volume over their course. like to adulthood. So,
Do they have anything equivalent to yolk?
You mean in the C. elegans embryo?
Yeah.
I don't think so.
So they may not increase in total. The total volume may not increase compared to the egg.
Yeah.
But that's not true in any yolk-bearing embryo or placental animal.
Right.
Okay, so it would be an interesting thing to determine.
Yeah. Yeah. So I mean, there are ways we could estimate the volume, and that's actually there's a set of interesting questions. I know people have kind of taken done segmentations of cells around the membrane and tried to, but I don't think people have actually looked at the question of volume distributions and things like that. I mean, there's been some work around the edges of that.
Volume versus time to see if they're growing.
Yeah.
I mean, they could grow at the expense of other cells that shrink, for example.
Yes.
If there's any, if the other cells are acting as a source of the equivalent of yolk. I mean, that's certainly what happens in yolk-bearing animals. The yolk cells are huge initially, and they decrease.
Yeah. Yeah, I know there's some asymmetric divisions locally in the tree where you get polar bodies. I don't know if that.
Yeah, that's an asymmetric. But then what's the volume of the two daughter cells? Does that increase?
Yeah.
And does it increase to retain the asymmetry, or is the asymmetry flattened out, so to speak?
Yeah. That's interesting.
Okay.
So that's what we call differentiation maps, and we can talk more about that later. I know Jia Hung was particularly interested in it, but so then there's this other paper on revisiting Waddington. So this is something that I know we come back to from time to time, and this is not maybe some of you aren't familiar with it, but this is I think this might be an accessible way to talk about it. So Waddington Landscapes, maybe I'll start with this one. So Waddington Landscapes are these structures that, if you're familiar with energetic landscapes or gradient descent landscapes, they're somewhat similar. It's the similar concept. I'm trying to find a picture in here of having a landscape that describes this process. And so in development, you have a process of differentiation of cells, differentiation of embryos, and you have this landscape that describes sort of the path of least resistance. And so, so let's see. I don't know if they actually have one of these, because they say it's revisiting them, but they don't say. Anyways, I wanted to show a picture of this landscape, but just imagine a landscape where you have the surface that's not flat, but has some undulations to it. And some of the undulations include valleys or canals. And so what they mean by canalization is that development tends to follow those canals from a point of a single cell embryo to the point of adulthood. And so this is a very conceptual map where, there are these different branch points down the landscape. So it's sort of like a lineage tree in that you have these different branch points. Development goes down one path, one canal, and then it can branch into other canals. And so you end up at the end of development at one point in this branch work of canals. So, it's like thinking about how many paths development can take. And so sometimes it can take alternate paths, and sometimes it sticks to the same path. And there are different reasons for that. But it's a conceptual model of how development kind of unfolds. And so in this paper, they're revisiting, they're re-examining Waddington. So they're looking at this idea of canalization, and then they're looking at this idea of genetic assimilation, which is where, I'm trying to remember the definition. It's been a while since. Okay, so why don't I go over the abstract? Over 65 years ago, Waddington demonstrated an ancestry of phenotypic plastic traits can evolve to become constitutive a process he termed genetic assimilation. So this is where you have plastic traits that become part of the developmental process. So you would have these things originally that were plastic from a source in the genome, and then you have the traits become part of the developmental process, and they're not plastic in the descendant. So this is what genetic assimilation is. It's assimilating this plasticity into the developmental process. Genetic assimilation evolves rapidly, assumed to be in large part due to segregating genetic variation only expressed in rare and novel environments. So this is where you have what otherwise phenotypically cryptic. So when organisms encounter these rare and novel environments, this is often the product of this, where you get this genetic assimilation.
Yeah, I've got the Waddington landscape on the screen. You want to
let me unshare my screen first.
Or
there we go.
I can see you.
Yeah, go ahead and share. Yeah.
Okay. Let's see.
Thank you, Harikrishna.
Share screen. Whoops. There we go. Share. Okay. Sharing now.
There we go.
Find it. Okay, I'll show it. You can talk.
Okay. Yeah. Nice works.
Okay.
Yeah.
Oops. Hello. There it is.
I think they have some things over it. You have to bring it to the front.
Have to do what?
Oh, yeah, there it is. Okay, I see it now. Thank you. Yeah, so this is the landscape here. This is like Waddington when he was writing his books in the early to middle 20th century. He kind of drew these by hand, and it was very conceptual. And this ball here represents an embryo. So it's in the form of a ball, and then you have these canals that come down. So there's this branch point here. So this ball's rolling down this canal, and then it either goes down this path or this path. And so then you have these these subsequent branch points that the ball can take, depending. In these canals are really shaped by genes and environment and their interaction. So this is a very complex process in this nice drawing. And so then, you get to the end and you get these 4 alternate paths for development. Where does the ball end up? One way to model this, if you're familiar with, I don't know, some of you don't know the game The Price is Right or the TV show The Price is Right, but there's this game called Plinko. And I know Dick has talked about this some in his books on Plinko, and how you drop it's a disc that you get up on a ladder and you drop it into this board of nails, and it kind of drifts down to the bottom, and you end up in a certain bin at the bottom, and you win a certain prize. This has its origins in what they call the Galton board in statistics, which is a similar thing, where you have this. It's a probability tool, where you have you drop something in the top and it bounces around, and it's probabilistic that it'll end up in any one bin. So this is that's a totally probabilistic example. But in development, sometimes things are probabilistic and sometimes things are deterministic. So this isn't going to just bounce around aimlessly. These things are going to branch off in different ways, but the end result is that you have these different categories that you can be.
Yeah. Bradley, I always had a problem with Waddington's picture.
Yeah.
If the ball represents the embryo, then the embryo becomes all one kind of cell. If it I don't know what else it could be, because there's no cell division here, and the embryo really should go back down all the paths.
Right.
Okay.
Yeah.
So, maybe it's a quantum embryo.
Yeah, maybe.
I never understood what the ball meant.
Yeah, I guess it's just a single state. I don't know, which is misleading because
Yeah, I mean, it's okay. It's a landscape, but the landscape for what?
Yeah.
What is traversing the landscape?
Yeah. And people have worked out mathematical models for the landscape in papers, like fairly recent papers, I guess. But they all kind of describe this landscape, something like what you might get with like a fitness landscape, or maybe even like a gradient descent landscape. In
any case, it does have a tree-like structure.
Right. And the idea in like a gradient descent landscape or is energy minimization, and in a fitness landscape and evolution, it's maximizing or minimizing fitness. And so in this case, they don't really have those criterion. Like you don't have these usual, you could call them optimization criterion. So it's hard to say like there is no optimization here. It's just kind of like you're going down one path or another. So okay. Well, I'm gonna go back to sharing my screen now.
Okay.
All right.
Okay, I have to stop sharing.
Yeah, you have to stop.
There. Okay.
There we go. All right, now let me share mine. All right, there we go. So that's what this paper, that's the context for this paper. And they re-evaluate this. And so they're actually using data. So they talk about how, despite previous work suggesting a substantial role of cryptic genetic variation contributing to the evolution of this assimilation, some have argued for a prominent role for new mutations of large effect concurrent with selection. So this is something that, it's a different mechanism. So if we go back to Waddington, he was less concerned by the relative contribution of CGV, or new variants, but aimed to test the role of canalization, which is this genetic assimilation. It's a very similar idea, because canalization is where you're taking these different paths based on what you're encountering in terms of plasticity. And this is also an evolved form of robustness. While canalization has been extensively studied, its role in the evolution of genetic assimilation is disputed, in part because explicit tests of evolved robustness are lacking. So they recreated Waddington's selection experiments on an environmentally sensitive change in Drosophila wing morphology. So this is the fruit fly. It's a model organism. They're looking at cross vein development in the wing of the Drosophila, and they show that they show a polygenic source, but not new variants of large effect are largely responsible for the evolved response, using both environmental manipulations and mutagenesis, which means they generate new mutations. There is no evidence for evolved changes in canalization contributing to genetic assimilation. And finally, there's a potentially what they call pleiotropic, or when you have a single genetic background, you have many different phenotypes that emerge, and fitness consequences in natural populations. It may not be entirely cryptic. So cryptic variation is where you don't necessarily know the source, or it's hard to characterize the variation. So if we go down to the figures on this, we have this example here of this is a Drosophila wing, and these are the cross veins you see here. And so, people have analyzed these quite a bit. They've done different things with image processing, and they've looked at like different aspects of the cross veins in the wing. It's a signature of developmental stress sometimes, or developmental perturbations, and people can learn a lot from that. From just you raise a bunch of fruit flies in captivity, you can control the temperature they're raised at. You can control other things. This graph shows where you have up selection, which is in blue, genetic assimilation, which is here, and down selection in green. And then it says alleles associated with this show strong response to selection. So up selection works for this trait. This is crossveins frequency, so there are more crossveins per square millimeter than in the ones that were down selected. So selection actually plays a heavy role in defining the crossvein frequency here. And then this genetic assimilation shows that this process here is delayed for about 17 generations and then matches what you see with selection. So this is, I guess they're doing some experimental evolution where they're raising different generations of Drosophila under different conditions and looking at them, and they're plotting out the results. This shows, this is a chromosome map. I don't think that's going to be of use too much here. And then this shows deletion line crosses average. This is pretty heavy duty genetics. I don't want to get into it too much, but this is more evidence. Looking for a nice graph might give us a little bit more information about the phenotype. So there are no differences in microenvironmental canalization or wing defect induced by rearing temperature. So oftentimes you'll see like wing defects if the rearing temperature of the eggs is at a higher level, or so when you raise them at a higher temperature, they end up with these increased number of defects. And so in this case, you didn't see any differences between macro or microenvironmental canalization or wing defect. And so this is interesting. It has a lot, it kind of shows that they're overcoming this effective temperature somehow. But that they're kind of mimic what you see with canalization. Environmental canalization. Okay, so that's this paper. I'm going to go to the next one. So I think that sets up this paper pretty well. And this is actually a lot more accessible and probably brings us home a little bit more. So this is, it's a feature that was in Cell. I don't know if it was in the journal Cell, but it's by Cell Press. And the question is how can Waddington Landscapes facilitate insights beyond developmental biology? And so this is like they asked a bunch of people, a bunch of scientists this question, and they wanted to get a sense of how can Waddington Landscapes go beyond developmental biology. So this is where, we're talking about embryos here, but maybe talking about other things as well that are related to maybe other biological questions or in engineering. So this person here, Nika Shakiba, she mentioned engineering beyond landscapes. So the Waddington landscape is often an intuitive framework to conceptualize self-fate, grounded in our innate understanding of the physical rules that guide balls rolling down a hill. So there's a connection here between gravity and the Waddington landscape. The hills and valleys of the landscape are molded by the cellular processor, the inner regulatory network of DNA, RNA, and proteins that govern cell state. So in this case, you can, and there are different ways you can characterize this, internal network of things. This is a lot of stuff going on here. DNA, RNA, and proteins. I mean, there are a lot of ways that they express different states. But, we often assume that it results in some single state and that it transitions between states. But maybe that's not a very good assumption to make. We also have microenvironmental inputs that act as forces that guide cells in their probabilistic journey downhill. So, we want to be able to break out of this metaphor and actually apply it to some problems. And so now, we can reimagine the landscape using predictive computational models and use it in mammalian synthetic biology and use it as a tool for designing landscapes that guide cell fate along prescribed paths. So in synthetic biology, oftentimes you're putting genes into a cell to get it to do specific things, like create some enzyme or create some protein. And so you can synthesize chemicals using these things, but you need to have a good understanding of how transitions work. when you modify the genotype or when you introduce it into a certain environment and what the output's going to be. So these landscapes might actually be useful for doing things like that. As an aside here, she mentions stochastic fluctuations, which is interesting because oftentimes molecular networks exhibit a lot of stochastic fluctuation. Sometimes this is a mechanism for robustness, which means it allows it to function in a lot of different settings where you might experience perturbations that might knock you off track. Other times it's just a product of it not being a deterministic thing. It's just something that happens irregularly, and it happens in parallel. So you end up with this signal that looks like it's stochastic. It bounces around an average, but it's really just this very highly parallel process. But we can if we understand that, then we have a better chance of sort of building control around it. Yeah.
A comment here. The language here smacks a bit of Stuart Kaufman's model of basins of attraction, where you could have a Markov process of transitions between them. The main problem I saw with Kaufman's model is that you end up with a whole network rather than a tree.
Yeah.
Okay, so the tree structure or the time development of the embryo, it does not become represented this way.
Yeah.
Okay. I mean, Waddington, at least presumably, going downhill meant because the time was going on.
Yeah. And for those who don't know about Stu Kaufman's model, that's actually a fitness landscape, or it's something similar to a fitness landscape. So, but the way that Stu Kaufman treats it, it's this basins of attraction model.
I think he wrote papers on using that as a model for differentiation.
Yeah, I think, he has a book, Origins of Order, that kind of goes through some of this.
No, you'd have to go back to his papers. I think they were on Drosophila.
Oh, okay.
It's a vague memory. Many decades ago.
To pull those up, at some point. So this next one is talking about quantifying energy landscapes, and we talked about this a little bit with respect to energy minimization and gradient descent, and how that's maybe similar, but it's not really the same thing. But in this case, this person is Chun Haley. He's arguing that again, it's a metaphor that there's this thing called energy landscape theory, which has been developed to study stochastic dynamics of cell fate decisions. So in this case, they're interested in energy as a mover, as a driver for some of these state changes in cells and some of their underlying gene regulatory networks. So again, in this case, in the landscape view, cell types are characterized by basins or attractors. So this is where those canals are now considered to be basins of attraction. And so I don't know if it suffers in the same issue that the Stu Kaufman work did. I imagine it's probably similar to that. But, this is something that could be applied to cancer and neuroscience, as well as developmental biology. And I've seen people do papers where they've looked at cancer where cells will transform away from their original fate and become cancerous, looking at that in this sort of landscape view. Although the landscapes are not Waddington landscapes in that they're not directional. They're kind of like these, nondescript attractor landscapes where you have a bunch of basins of attraction that are just kind of in space, distributed in space because they're different states. And so they don't really they're not linked to any sort of process directly. I mean, the distance is process. It moves from one place to another. But this is again, something that we think about in terms of energy and energy minimization and energy function. So in neuroscience, Hopefield pioneeringly proposed the concept of energy function to explore computational properties of neural circuits, such as associative memory. So maybe something like a spiking neural network model might be integrated with Waddington Landscapes, and that can drive some insight. So Jordi Garcia Ojalvo it says research evolution. That's his answer. So over 20 years ago, the late Sidney Brenner said that physics is the art of the optimal, while biology is the art of the satisfactory. And so then Conrad Waddington has been suggesting that through his metaphorical landscape, developing organisms are also skilled optimizers, finding surface minima as they self-organize. So this is something where they're kind of making the connection more explicitly to optimization. So, I said that the Waddington landscapes aren't really about optimization, but they can be about optimization. You have to be a little bit more specific in how how they map to the developmental system. You can't just use it as a metaphor. But the idea here is that a single dimension can account for the state of a cell as decisions are made is clearly insufficient in light of the current high-throughput single-cell surveys. You should also not ignore the fact that cells actively carve out their physiological landscape as they implement their decisions. So these channels aren't deterministic. They're kind of pushed. They're kind of carved out as the cell moves along these state pathways. But so this just talks about how if we incorporate more of these things that we know about the biology, it may be a tool for optimization or understanding optimization in a biological sense. This is self-fate reprogramming. I've talked about this with respect to cancer cells, but people have also used this to understand what happens when you reprogram one cell to another type of cell. So we can reprogram skin cells to neurons or to stem cells. And where we can program stem cells to neurons or muscle cells. And all those things can be characterized using a kind of a landscape metaphor, understanding sort of different transitions and their time points and their multidimensional structure of these processes and put them into a landscape. So this is kind of moving from this Waddington landscape to something of a complex network control strategy, which is where you use a landscape to characterize some of these changes in multidimensional space. Again, this is kind of moving away from the Waddington landscape and into some of these other tools and kind of interacting with them. So this person here, Kiran Patel, he argues for a geometry of intuition. And so this is kind of talking about how we know now about molecular players and networks, which we didn't know back in Waddington's time. We know a lot more about their dynamics and we can measure a lot more. But yet the quest to distill the discovery and dynamics of biological systems into its basic organizational principles is far from complete. So this is where geometric perspectives and metaphors can play a fundamental role. So we know, for example, like I've mentioned, that we have these convex spaces that can describe optimization. We have stability theory, which come in the form of saddle points, which are points that are in between two stable states, and your system can go into one stable state or another. And energy barriers from enzymology. And so there are all these tools from different places. And so this may be a way to integrate using the power of this sort of intuitive geometry to bring all these together into this context of the whole cell or the whole embryo understanding. And so this has links to complex systems theory, which we've also talked about. But this just basically, it helps us structure our debates about what the basic principles of biology are. And then, we can also look at attractors as linking directly to data. And so Alexandra Walczak, who is at French University, sort of argues that you can actually look at data in this way as well. So, we build, so from her perspective, cell differentiation pathways have diverse implementations that lead to a set of finite states. But as these two authors have shown a few years back, it can also simplify molecular description and lead to quantifiable experimental predictions. These models also build on the intuition we see in many living systems that phenotypic states are stable and reproducible, but the molecular and genetic implementation can vary. So this idea of buffering or of assimilation actually is something that we observe in the data, but it's something that we need to have an intuitive model for at the level of a Waddington landscape. And indeed, Waddington talks about this, but it doesn't have the data to show that, how this unfolds. And so there's a linkage here between a lot of the data that's been collected out there and bringing this back to a landscape model. So in protein folding, the idea that proteins differ from other polymers by having a funneled folding landscape, which helps them avoid misfolded structures, explains how they can fold in a finite time. And so this is something you can also see in protein folding, where you have these challenges of this process, and it ends up in this sort of state space from a large number of possibilities. And so, using examples from the data that we have is actually quite useful. Then there are other examples. I'll kind of go through the rest of this, just kind of like pointing out the applications. So this is human microbiome landscapes. So applying microbiomes to these kind of landscapes. Learning from natural variation, which is where you're learning, observing natural variation. Talks about canalization being something like the wild type, which is like a normal genotype instead of a mutant genotype. So, if you have your mutants actually generate more channels during canalization. So you can learn a lot about natural variation by these models by adding in canals and branch points. And so this is something that people might be interested in doing. Single cell dynamical landscapes. So this is very similar to what was just mentioned, except that you want to see what single cells are doing over time and where they end up in terms of their state space. And this is really driven by advances in single cell RNA-seq. So this is a specific type of data that looks at gene expression, at large scale gene expression. And you can unpack a lot of this data using dynamical systems methods. And then again, single cell dynamical landscapes, brain attractor landscapes, which then goes to the brain and some of the things going on there beyond the evocative picture. So this is actually talking about this mathematical framework and visual metaphor connection. So, now we're talking about doing higher order mathematics using landscapes, and actually landscapes can capture things. So this picture describes a dynamic system followed by homeoresis, which represents the return to a stable trajectory, a set line, after perturbation. And this is unlike homeostasis, which returns to a set point or a particular state. So this is something that, now we're talking about regulation and some of the theories of regulation that exist. And so this is now moving towards sort of theory building. So that's all they have in that paper. I think that was a nice paper, or a nice tour of what people are thinking about when they think about some of these developmental processes and some of these tools, or conceptual tools we use in development. There we go. Did we have any questions about that, or comments? Okay. Well, thanks for meeting. And we won't be meeting next week. It's a holiday in the U.S. We'll be meeting the following week. For the GSoC students, I want them to give updates in the Slack. I think we can do that. And then the following week, maybe we'll have some nice demos to show from your several weeks of hard work. The first evaluations for GSoC students is at the end of July. So that's not that far off, but I want, I don't usually make it hard for people. I try to make it easy for people. So it's, I just want to make sure that you're making progress on your projects. And so if you have any questions, let me know. But it's usually just a formality. I think all of you are doing well. But just keep that in mind, because you'll actually owe them an evaluation, or you have to, you have to give feedback or something. But it's largely on me. But there's a deadline for that at the end of July. But otherwise, thank you for attending, and talk to you online.
Bye.